All right, so this next lecture, our third lecture, goes over the cell structure. So uh, in this, we're starting off with the cell membrane. So the cell membrane, it's also called the plasma membrane or the plasma lemma, right? This is a biological membrane that's separating the interior of the cell from the outside environment. So therefore, the internal and external environment are different. Okay, in terms of what we find, it's the constituents and the chemical makeup also of the fluids that are present. So it's protecting the cell from the surrounding. That's another property of this plasma membrane or cell membrane. And what it is made up of is the following. The plasma membrane, it is made up of phospholipids. It's made up of some cholesterol molecules. There's lots of different type of proteins. Some of them are called carrier proteins. Others are channel proteins. Some of them are glycoproteins. Some of them are integral proteins. Some of them are peripheral proteins. So we'll be taking a closer look at all these different type of proteins that we find within as part of the plasma membrane. So again, this by use of these plasma proteins is how it's mostly able to exhibit some of these permeability properties the selective permeability property that the plasma membrane has. So again, by selectively permeable, we're referring to the plasma membrane's ability to allow a certain substance to enter and or exit for that matter, and other things it won't allow to come in or go out of the cell. The cellular membranes, okay, so they call this a fluid mosaic of lipids and proteins. So what does that mean? So first of all, again, these phospholipids are the most abundant lipid in that plasma membrane. Phospholipids are said to be amphipathic molecules, meaning that they have both a hydrophobic as well as a hydrophilic region. And this fluid mosaic model, what they're referring to is that the membrane, it has this fluid structure with patterns, okay, of proteins that are embedded within it. Okay, so again, you have a backdrop of phospholipids, and embedded within these phospholipids, you have these proteins. Okay, so this is where that mosaic comes from. So membranes, they've been chemically analyzed, and we found that that they're composed of both lipids as well as proteins. Scientists that are studying the plasma membrane, they reason that it must be a phospholipid bilayer. So what they did is they did this freeze fracture study of the plasma membrane, and this freeze fracture study, it supported this fluid mosaic model. And we'll take a look at this freeze fracture study, I believe, in the next slide. So again, this freeze fracture, this is a specialized, it's a technique, a preparation technique that it splits the cell membrane along the middle of the phospholipid bilayer. So let me show you what happens. If you look over here, essentially they take a cell and they freeze it. Now, once this plasma membrane freezes, what happens? It's not moving anymore, right? Things, whatever that are its constituents, they're frozen. They're stuck. They're frozen. I don't know another term to explain that to you. So they're paralyzed. They're not able to move anymore. Now, once that happens, now what they do is they take a knife and they slice it. They slice that plasma membrane. They cut it, and then they observe the properties of it. So this was done in 1935 by Hugh and Davison, along this person named Hugh Davison and his partner James Danieli. And so they proposed this sandwich model, which the phospholipid bilayer lies between two globular proteins. This is what they thought, okay? Dyson and Daniel, right? So they thought, we had proteins that acted as the buns, I guess, of this phospholipid bilayer. they weren't correct. They were inaccurate, right? They were wrong about that. So later studies, they specifically, studies, they found a problem with their idea of, with Davison and Danieli's model of this, the sandwich model. They're like, oh, something doesn't seem right. So in 1972, this guy named S.J. Singer and his partner G. Nicolson, they proposed that the membrane is a mosaic of proteins that you find within this bilayer. Okay, and they said that the hydrophilic regions were exposed to water. That's it. That's what they came up with. And this is, at that point, they ended up doing this freeze, Singer and Nicolson, they ended up doing this freeze fracture study. So that's who's credited with for that. So as far as the fluidity goes, okay, the fluidity of the membrane. So the phospholipids in the plasma membrane, they're able to move around within that bilayer very easily. They're constantly moving back and forth, laterally, side to side, constantly. They're always on the move. So that's why we say it's in a fluid state. And this is another reason why Singer and Nicolson, they had to freeze before they were able to study it properly. So most of the lipids and some of the proteins, again, the vast majority, most of the proteins, they're drifting laterally. The plasma membranes, the lipids, again, lateral movement. Now, sometimes it happens very infrequently, but it may go back and forth as well. Okay, it may flip-flop transverse across this membrane. It's very rare, but it does happen. The vast majority of movement occurs laterally. So the phospholipid, it has both a polar and hydrophilic regions. In other words, they're saying that they have a charge. So it has a polar, okay, and a hydrophilic end, and then the tail is non-polar and hydrophobic. There's no charge there. So they could also be saturated or unsaturated, right? So if there's a double bond, it leads to a kink in the shape of the tail. And again, we find this, so in between the phospholipids, we have the cholesterol molecules that are dispersed as well. So cholesterol molecules we find in between if you look in between the phospholipids, we have the cholesterol molecules. So you can see one over there. So again, and these cholesterol molecules, they help manage the distance between the phospholipids. So again, they're very important. Cholesterol molecules are important. They're not too much cholesterol is bad for you, but we still need it. It's important for survival. So again, if you look over here, this is this membrane fluidity that we're talking about. So they're constantly moving back and forth, not back and forth, side to side, lateral movements. Okay, look at this, 10 to the 7 times per second. This is how enormously fast, we have movement taking place. However, going back to forth, again, transverse movement, that's very rare, as I said earlier. Perhaps only once a month do we get that movement taking place. So now, the other thing I told you was, their shapes are a little bit different. The phospholipid, the tails. So this is the hydrophilic region, the heads, and then this is the hydrophobic, the tails. Now, what we found is this. So temperature and membrane composition. So the temperature does have an effect on the fluidity. As temperatures cool, membranes, they switch from a fluid state to a solid state. So cooling made them less immobile. In other words, they became more solid. The temperature at which the membrane solidifies was observed by Singer and Nicolson. So membranes that are rich in unsaturated fatty acids are more fluid than those that are rich in saturated fatty acids. Okay, so unsaturated, these guys over here, they're going to be more fluid, okay, than these ones over here, which are saturated. Membranes, they have to be fluid in order for them to function properly. And again, they're usually about as fluid as salad oil, the oil that you put in your salads. This is their fluidity. Now, the steroid cholesterol, it helps stabilize these. Right? This is so cholesterol is a type of a steroid, and again, it has effects on this membrane fluidity at different temperatures. So room temperature consistency, again, it's about the oil that is, so vegetable oil, right? This is, and this is ideal, okay? At higher temperatures, right? Higher temperatures, it causes the cell membrane components to move further apart from one another, okay? Higher temperatures, the cell membranes, they start to move further apart. So when this happens, now they're too fluid, and it causes the membrane to start to break apart. It causes it to disintegrate. At cooler temperatures, the phospholipids, they clump together. They come together very closely. They huddle up, and it makes it much more difficult for things to come in. So now it's too rigid. So it makes things very difficult to come in or even go out, for that matter. Cholesterol, however, is very stable, and it helps the phospholipids stay at an optimal distance from one another. This is their key thing, right? They help stabilize the distance between the adjacent phospholipids.
Evolution-wise, adaptation, and again, we said that again, the variations in the composition of the lipids that we have that are part of the cell membrane, it is thought to have evolved due to environmental conditions. Okay, and again, environmental conditions specifically we're referring to as the temperature effects, the temperature changes that have taken place over the course of great amount of time. That again, these components start to evolve in organisms based upon where we find them. They have different optimal temperatures that they function at. All right. So that was the last slide. Now we can kind of go up. So the membrane, again, the membrane is a collage, okay, of different proteins, and they oftentimes they're grouped together, okay, and they're embedded within this fluid matrix of the lipid bilayer. Proteins determine most of that membrane-specific functions. Okay, don't forget that proteins, they end up determining the vast majority of this membrane's specific functions. When we talk about peripheral proteins, again, we say that the peripheral proteins are bound to the outer surface of that, the outer or inner surface of that membrane. Okay, peripheral proteins, therefore, are bound to the surface of the membrane. Could be the outer membrane or it could be the inner membrane. Contrast that to the integral proteins. The integral proteins, they are going to be penetrating the hydrophobic core. In other words, in this picture, when you look at it, they go from one side of the phospholipid membrane to the other side. So again, you have the outside of the cell, which is in blue, and then the inner part, that's going to be the cytoplasmic side of the cell. All right, that's the part in beige. And again, when you look at this protein, you can see the different parts to it. First of all, you see that this protein, it's in an alpha helix arrangement. And then you can see that the N terminus and the C terminus of this protein as well. So integral proteins, again, they penetrate the core of this plasma membrane, okay? Now, not all integral proteins go from one side to the other side. Not all of them go from one side to the other. Some of them, they'll just go to, they'll be stuck only halfway within that plasma membrane, okay? Or meaning only on one side of that plasma membrane that they'll be put into, okay? But they're still embedded within this. They still penetrate, again, to the core, right? The hydrophobic core. In transmembrane proteins, okay, now these are the integral proteins that go from one side of the phospholipid to the other side of the phospholipid. Okay, so they span both sides of the phospholipid membrane. Okay. The hydrophobic regions of this integral protein, okay, they're made up of one or more stretches of non-polar amino acids. Okay, it's non-polar amino acids, and again, often they end up getting coiled into what we see, the structure that's formed over here, which we call this alpha helix. Okay, All right, so when you look over here, look at some of the membrane protein functions that you see. So these ones over here, for example, they're transporting specific particles or some ions in this case, okay? So these are transport proteins. Others, they end up forming channels where things just move in and out. Others, they recognize a specific particle and then they allow it to enter. Okay, so transport proteins, they can be more specific to what they're allowing in and out. Other proteins, they function as enzymes, okay? They have special receptors that facilitate the movement or, again, reaction of certain chemicals with other components to it. Other proteins, as an example of, this is an example of, for example, a G protein, for example, it would be for signal transduction, okay? In other words, it allows they act as messengers, okay? They start the process of sending or creating a message that's going to end up changing the functioning of that cell. All right, so that's that would be signal transduction. So again, they can function, the proteins, they can function as transport of substances. They can function for enzymatic activity, and also they can function as for signal transduction. All right, next there's the cell-to-cell recognition, okay? Cell-to-cell recognition as well. So here's one cell, the picture all the way on the left, all right? And there's another protein on the other cell. So it allows these two cells to communicate with one another and recognize one another, right? They can also help connect different cells, so intercellular joining, right? They can also act as structures to where other components within the cell attach to as well, right? Within or outside of that cell end up attaching to. So they end up being attachment points to the cytoskeleton as well as for the extracellular matrix, right? Now, cells, they recognize one another by binding to surface molecules. And often they will, they contain carbohydrates on the extracellular surface of that plasma membrane. The membrane carbohydrates, they could be covalently bonded to lipids. And when that happens, so they could form glycolipids. Okay? More common than the glycolipids, they end up forming glycoproteins. Okay? So glycoproteins are far more numerous than glycolipids. Carbohydrates on the external side of the plasma membrane, they will vary according to species, individuals, and even cell types within an individual. Okay, so again, huge amount of variation. Membranes, they have a distinct inside face and a distinct outside face. This asymmetrical distribution of proteins, lipids, and again, associated carbs in that plasma membrane is going to end up determining when that membrane is built by the endoplasmic reticulum and Golgi apparatus. Okay, so the ER and the Golgi apparatus, it helps build this membrane also and maintain it. This is also something important to realize and understand. It's going to be producing different parts of it. Now, a cell, it has to exchange materials with its surrounding, right? And this process, as you can imagine, ends up being controlled by the plasma membrane, right? We said that very early on. Okay, so the cell, it has to be able to exchange materials with its environment, with the surroundings, right? And the plasma membrane's job is for that. Now, in doing so, we see that the plasma membrane is selectively permeable. Selectively permeable means, again, it has the ability to regulate the molecular, what molecules are able to, or molecules or atoms are able to come inside and go outside. Okay, so again, it's protein. The plasma membrane, it is controlling this. Okay, what's moving in, what's moving out, right? And this moving in and moving out, we say is termed selective. It's selectively permeable. Okay, selective permeability is what is picking what is able to come in and what is able to go out, or what not to allow in. So non-polar molecules, okay, we're talking about hydrophobic, non-polar molecules, like hydrocarbons, they're able to dissolve in the lipid bilayer, and they can pass through that membrane relatively fast, right? Polar molecules, however, such as sugar, they don't cross the membrane easily, right? Sugar has a hard time entering, which is why it needs help. And this is why our body produces insulin. Insulin essentially picks up the sugar molecules and it helps push them inside, deliver them inside of the cell. Okay. Okay, why sugar is a polar molecule. So slide number 16, okay, it's going over transport proteins. So again, as the name implies, transport proteins or transport is going to allow passage of substances across the membrane. Okay, however, what type of substances? This is the question. Based on, so this is going to be, it's going to allow hydrophilic substances across that membrane, okay? So transport proteins are going to allow hydrophilic substances passage across that membrane. Now, some of these transport proteins are called channel proteins. Okay, now so these channel proteins, they have a hydrophilic channel, right? So this hydrophilic channel, therefore, will allow the passage of what? Hydrophilic molecules or ions. Okay, so again, they use this channel. Channel proteins that are called aquaporin. Okay, if you know what aqua means, A-Q-U-A, aqua means water. Okay, porins means again a pore, passageway. So this aquaporins, these channel proteins allow the passage of water. All right. Other transport proteins that are called carrier proteins. Carrier proteins will bind to molecules and then they change their shape to move them from transfer protein is, again, so transfer proteins are specific for the substance it moves. Okay, transfer proteins, they're specific for the substance that they move. All right, now diffusion. Diffusion is the tendency for molecules to spread out evenly into the available space. Okay, this is what diffusion is. Diffusion, again, you're talking about, well, there's different, so again, movement of substances evenly into the space, this is what diffusion is. So now, although each molecule moves randomly, diffusion of a population of a molecule, they could be directional. Now, at equilibrium, many molecules will cross the membrane in one direction as in the other. Okay, that's what in dynamic equilibrium. Whatever moves in one direction, the amount of molecules, the same will move in the opposite direction. So if you look at these pictures over here, the picture all the way on the right, on the bottom, for example, or even the one on the top, what they're showing is, look, there's 2 purple or 3 purple on one side, and there's 3 purple on the other side. There's 6 yellow circles on one side and 6 on the other. So if one yellow goes from the left side to the right side, then another one, yellow, will go from the left to the right also. So you have equal, okay, number of movement. That's equilibrium. Osmosis essentially is water. Well, actually, I'm going too far ahead of myself. Let me wait till we get to the next slide. Next slides. So again, if you look over here, what's happening? You have the picture on top. We're talking about net diffusion. So again, you have this water and then you have a membrane. Okay, these are the dashed lines. So essentially, when you put these molecules, and it could be anything. In this case, you're just saying that it's a molecule of a dye. So what's happening over here? There's on one side, this is the picture all the way on the left, on the top. There's not any molecules on the right side, and there are some on the left. So what's going to end up happening is the molecules on the left side, they start to move to the other side. Okay, so again, you're having net diffusion take place. These molecules will start to go to the other side. So again, substances, when we say they diffuse down their concentration gradient. Okay, don't forget, substances, they're going to move down their concentration gradient. This is the region along which the density of chemical substances either will increase or they will decrease. And no work needs to be done in order to move these substances down their concentration gradient. In other words, that means no energy is required, right? No energy is required. So the diffusion of a substance across a biological membrane is passive transport. Again, no energy is required. Diffusion of substances across a living membrane is passive transport. No energy is required by that cell to make it happen. All right. Now we look over here. What's osmosis? Osmosis is just the diffusion of water across a selectively permeable membrane. Okay, osmosis is just the diffusion of water across the selectively permeable membrane. And water diffuses across that membrane from regions of lower solute concentration to the region of higher solute concentration until the solute concentration is equal on both sides, right? So when you look at this photograph over here, what's happening? We see that on one, let's look at the green balls on the left side in the picture on top. There's 1, 2, 3, 4, 5, about 6 balls on the left side. And on the right side, we have 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 balls. So on one side, a lot of balls, and the other side, not as many balls that are present over there, these green balls that are there. All right, so water is going to go from where the balls are more concentrated. It's going to want to move over there. Okay, so when you look at this picture on the right, what ends up happening is the water on the left side starts to decrease. Why? Because again, there's more solutes that are present on other sides. So again, we said that water is going to go from area where it's going to move to areas where it's less concentrated. All right. So again, it's going from this area on the left to the right. Now the amount of water per solute. So in other words, before we talk about solute, let's talk about what does a solution consist of? What parts are there to a solution? Solvent is going to be part of the chemical makeup. That's going to be much higher in quantity, whereas the solute is going to be part of the chemical makeup. That's going to be much less in quantity. So in this case over here, we're talking about solvent and solute. A lot more water is present, so we say the water is going to be the solvent. The green balls, the green dots, these are the solute. Okay, so solute, solvent. Now, when you combine solute and solvent, now we call that a solution. Okay, so solution is a combination of both solute and solvent. All right. So in this picture here, essentially we're saying that again, this water is concentration. It ends up, the water is going to go from where the solute is less to where the solute is more. Okay. So on this picture on the right side, we see that the solute concentration ends up being equal on both sides now, right? Now we go over here. So look, this is what happens in a, let's talk about water balance in this, in a cell, right? And they use the example of a red blood cell. Now, tenacity tonicity. Tonicity. This is the ability of a surrounding solution to cause a cell to either gain water or to lose water. Okay, that's what tonicity is. The ability of the surrounding solution, okay, that's gonna have an effect of that cell to either take in water or to push water out. That's what tonicity is. Tonicity is. So what would happen to a red blood cell if we put it into an isotonic solution? Okay, so let's just talk about, this is salt water, all right? 0.9%. This is the salt concentration of whole blood. Okay, 0.9%. All right, this is the concentration of salt in our blood. If you were to take in our whole blood, if you were to take a red blood cell and put it into a glass or a beaker or a test tube where the salt concentration was 0.9, nothing would happen to that cell. The cell would be perfectly fine. It would stay in that same state. In other words, we say that it's going to be in a flaccid state. Okay, say it's flaccid. It doesn't shrivel up. It doesn't get fatter. It just kind of stays more or less the same. Okay, it stays in this flaccid state. And again, we call that solution, we say it's isotonic. Okay, so in an isotonic solution, the solute concentration is the same as that inside of the cell. Okay, so again, in an isotonic solution, we say the solute concentration ends up being the same as inside of the cell. So again, the salt concentration inside the cell is also 0.9, this red blood cell, and then also outside is also 0.9. All right. So in other words, the amount of water that's going into the cell is going to be the same amount of water that's going outside of that cell. So in other words, everything is good. Cell stays flaccid. We have an isotonic solution. Now let's look at the one all the way on the right. It's hypertonic solution. Okay. In a hypertonic solution, the solute concentration is going to be greater than inside that cell. So in other words, the cell is going to be losing water. So what's happening over here? Now we said that this cell, first of all, whole blood, it's 0.9% salt. Right? That's what it likes. Same thing with the red blood cell, 0.9%. That's what it has. Now, if we were to put the salt, let's just say in 2.7%. Okay, so 2.7%, that would be what? 3 times the norm. So salt concentration. So what's going to end up happening is this. The amount of water, the amount of salt that this water has is very high, 3 times the amount. So as that water, the amount of salt that this water has, right, that the cell is inside of, is 3 times the normal. So as water enters the cell, three times the amount of salt also goes inside the cell. And this cell is like, whoa, that's too much salt. We need to get rid of it. So as this cell is pumping out that salt, along with it, a lot of extra water also starts to get pushed out as well. Okay, and when that happens, the cell starts to get, it starts to shrink. All right, and we call that again, the cell becomes plasmalized, okay, or it creates. All right, it creates or it will plasmalize. Okay, that's the state it's going to be in. So in other words, the cell just shrivels up and it's going to die. All right. In other words, what are we having? Dehydration take place, right? The water amount is being pushed, too much water is being pushed out. It'll start to dehydrate. Now let's go all the way to the left side. The first picture all the way on the left. Now, if you take that red blood cell and you put it in a hypotonic solution, now we're saying that hypo means less, right? So the salt concentration, it's going to be, let's say it's half, so not 0.9%, but 0.45%. That's half. So what will happen at that point? Now, the amount of salt that's present in this water, right? It's half of what is normally half of what is present inside the cell. So the cell's like, whoa, there's not enough salt. We need more salt. So let's suck up more water. So as more water is coming in to get that salt to help maintain that 0.9, too much water is coming in. So now the cell becomes very, it starts to get big. It gets plumpy. So we say that it is turgid, right? And when that happens, then what's going to happen? When it becomes turgid, eventually, as more water comes in, the cell blows up or it lyses. Okay, so cell dies over here as well. Cell dies here, cell dies there. In hypertonic solution dies, hypertonic solution will also die. So osmoregulation, hypertonic or hypotonic environments, again, they create an osmotic problem for the organism. Now, osmoregulation, this is, again, as the name implies, regulating osmosis. Okay, it's a control of the solute concentrations and water balance. Okay, and this is a necessary adaptation for life in these environments. So again, when you look at this protist, this is a paramecium. Then it's in a it's hypertonic to its pond environment, and it's got these vacuoles, okay, that act as pumps that helps push that water in or push it out. Well, in this case, if it's hypertonic, it's going to help push out that water efficiently and effectively so it can survive. So again, this is an adaptation that's built. So again, I told you about turgid, flaccid, and plasmosis. So again, cell walls, it's going to help maintain this water balance. So if you take a plant cell in a hypotonic solution, okay, hypotonic solution, same thing as our red blood cell, that it's going to end up swelling up. Okay. So what do we say? That it's turgid, right? It's too firm, turgid. Now, if the plant cell and its surroundings are isotonic, then there's no net movement of water into. There is a net movement of water in and out of the cell, right? What's going in is going out, and the cells become. It's going to end up becoming flaccid. In a plant's case, it'll be a little bit. It'll be limp because there's net movement, isotonic. Now, in a hypertonic environment, that plant cell is going to start to lose water. Eventually, again, the membrane it's going to pull away from the walls. And again, you end up having plasmolysis take place. It will wilt. Okay. All right. We talk about facilitated diffusion. Now, in facilitated diffusion, these transport proteins, they speed the passive movement of molecules across the plasma membrane. Right. Facilitated diffusion. Right. Transport proteins, they speed the passive movement of molecules across the plasma membrane. So channel proteins, they end up making corridors that allow specific molecules or ions to cross that membrane. And channel proteins, as I think I told you earlier, channel proteins, they include alkoporins. And again, alkoporins, they provide for facilitated diffusion of water. Right? Again, no energy is needed. Ion channels that open or close in response to a stimulus are said to be gated channels. So a calcium channel, for example, is a gated channel. Okay, calcium it's a voltage gated channel because a current ends up opening up a calcium channel. So yeah, remember that. Chiral proteins, they undergo subtle changes in shape that translocate the solute binding site across that membrane, right? So chiral proteins, they're going to undergo small changes in their shape that's going to move the solute binding site across that membrane. So you can see that's a picture on the bottom. That's that of a carrier protein. Something comes, it binds, okay, then the shape changes, and then it ends up emptying that solute to the other side. All right. Whereas the picture on top, again, this protein just ends up making a channel for whatever these round circles, these balls are to pass through. So facilitated diffusion, again, facilitated diffusion is still passive because the solute moves down the concentration gradient. And this doesn't require any energy, no energy. Some of these transport proteins, they can move solutes against their concentration gradients, right? Active transport, it moves substances across their concentration gradient. Now, active transport, it is going to require energy. Active transport requires energy. And again, this energy, it comes in the form of adenosine triphosphate, ATP. Active transport, again, it's performed by specific proteins that are embedded within that membrane. Active transport also allows cells to maintain concentration gradients that differ from their surroundings. So a very good example of active transport would be the sodium-potassium pump. Okay, and this is one of the type of active transport systems, sodium-potassium pump. What does it do? It's moving sodium and potassium. So again, diffusion, what happens? Right? Things just go from one side to the other, right? They just move down their concentration. Facilitated diffusion, same thing. Things go from one side to an area of lower concentration to other concentration, but they happen with the help of a protein. Still no energy is needed, right, for facilitated diffusion. Now, active transport, however, okay, active transport, you have proteins that are moving things across the concentration gradient. However, so if you look over here, they require energy. Let's just keep it at that. They will require energy. Now, how ion pumps maintain membrane potential? So again, a membrane potential is the voltage difference across a membrane. A voltage ends up being created by differences in the distribution of positive as well as negative ions across a membrane. When you have two combined forces collectively that are called electrochemical gradients, they drive the diffusion of ions across that membrane. A chemical force, again, chemical forces are going to be the ion's concentration gradient. It could be an electrical force. And again, electrical force is going to be the effect of that membrane potential on the ion's movement. When you have electrogenic pumps, okay, an electrogenic pump, this is a transport protein that generates voltage across that membrane. So again, a sodium-potassium pump, this is a major electrogenic pump that's found in animal cells. Okay, so again, sodium-potassium pump, this is an example of an electrogenic pump. The main electrogenic pump of plants, in bacteria and fungus, or fungi, I should say, right? Fungi, plural. It would be proton pumps. Okay, proton pumps for bacteria, plants, fungi, and for us, you know, in animal cells, we have sodium-potassium pump. This is a major one, the huge one. So you can understand that these electrogenic pumps, they help store energy that can also be used for work later on, for cellular work. Right? This is one of their functions, purposes. So cotransport, okay? Cotransport occurs when active transport of a solute indirectly drives the transport of another solute, of other type of solutes, okay? Plants commonly they end up using the gradient of hydrogen ions that's generated by proton pumps to drive active transport of nutrients into the cell. So again, when you look at this, what are we seeing? We're seeing this proton pump is being pushed out. Again, it requires energy, right? Proton pump is pushing out protons or hydrogen atoms, right? And then you have the sucrose. So as this proton pump is pushing out this, it ends up making, it ends up helping this sucrose protein to bring sugar in. The hydrogen, as it comes through, it gives the energy that's needed. It'll draw it in. It helps drive indirectly, it helps deliver the sucrose. So again, active transport, it is requiring energy, it's pushing out the protons. The sucrose hydrogen co-transporter, okay? As hydrogen is coming in, it also helps bring in sugar. If hydrogen is not going to come in, then sugar cannot come in. Okay, so again, this is a co-transporter. Something else that's coming in helps bring something else. That's essentially it. It transports two different things. Okay, but both are required. One is required for the other to come in. Co-transport. Again, if you look over here, look at the shape of this part, this recognition site. It's circular, and as the recognition site over here, it takes the shape of this sucrose molecule over here. So this part's going to come in and fit over here to allow for the sugar to come in, to enter. All right. In this slide, so small molecules and water, they enter or leave the cell through the lipid bilayer or via transport proteins. Right? Small molecules as well as water, they enter or they leave the cell through the lipid bilayer or through transport proteins. Large molecules like polysaccharides and proteins, they can cross the membrane in bulk by vesicles. So bulk transport, it requires energy. Now in exocytosis, the transport vesicles, they migrate to the membrane, they fuse with it, and then they release their contents. A lot of secretory cells, they use exocytosis to export their products. The opposite of that would be endocytosis. Now, in endocytosis, this is a cell taking in macromolecules by forming vesicles from the plasma membrane. So exocytosis, you're pushing substances out. Okay, bulk transport. Okay, again, it requires energy to push things out. Now we're looking at endocytosis, not pushing things out, but taking things in. Okay, and we say that again, it uses the plasma membrane to form vesicles to take things inside. Endocytosis is, again, when you think about it, endo coming in. So it's the opposite of exocytosis. So it's a reversal, right? However, it does require different proteins. Now, there's three types of endocytosis. There's phagocytosis, which means cell eating. Phagocytosis, cell eating. So you're taking in large amounts of solid structures, okay? Big molecules are taken. There's penocytosis. Penocytosis means cell drinking, cellular drinking, and you're taking in large amounts of liquids of some type. Then there's also receptor-mediated endocytosis. Again, receptors, receptors are needed for whatever substance to be able to come in. So again, you need to have protein receptors that are going to bind to another substance to allow for that substance to enter. So now in phagocytosis, a cell eats, okay, or engulfs a particle in a vacuole, right? That's going to be, so this is a food vacuole. This is phagocytosis. The vacuole then fuses with a lysosome later on, and it digests. It's going to digesting that particle. In this picture on the right, in penocytosis, molecules are taken up when the extracellular fluid ends up being, again, gulped into these tiny vesicles. It just kind of come in, the plasma membrane forms around it, and then it ends up coming inside. Right? Penocytosis, water also is coming there. receptor mediated, right? In receptor mediated endocytosis, you end up having these proteins on the outside of the cell. So these protein receptors. These protein receptors, they're very specific as to what they can bind with and connect with. So again, if you notice here, you got purple triangles and green circles, all right? These green balls. The green balls are not going to end up coming and attaching to these Y-shaped structures, right? Because they don't fit. So these green and purple structures are referred to as ligands. So receptor-mediated endocytosis is, we say that is ligand specific. In other words, the ligand is going to be a molecule that binds specifically to a protein receptor of another molecule, right? So again, ligand, any molecule that binds specifically to receptor sites of another molecule. That's what you're seeing over here. These purple dots, they're going to end up coming over here. Right? And these purple triangles, they'll attach to these receptors. Okay. Now, once they attach, then the plasma membrane, it forms a vesicle around them and it takes it in. You can see in the picture in the bottom. Now, these purple things are inside the cell. And then along with it, these green things, they also sometimes they'll also end up being pulled in. Once they're inside, then again, if this is something bad, for example, then these lysosomes would then end up eating it up. Lysosome would fuse with this part, and then it would eat up, break down, and break down this part, disintegrate it. All right, so that is it for this lecture. We are done for today for this unit. So be sure you review it.